Autoantibodies are a type of antibodies which are proteins produced in our body normally in defense for virus and bacteria. But a subtype of antibody, the autoantibodies, end up produced targeting our own cellular molecules or structures. And this is basically related to our immune system evolved over many years to fight against bacteria and viruses. In order to be effective, it has to have a very diverse mechanism to recognize all foreign structures from the chemical moiety point of view. And in order to do that, there are chances that it will also recognize self molecules, and those are autoantibodies that we are talking about.
What are some of the diseases caused by these autoantibodies?
These are under the general category of autoimmune disease. There are some autoantibodies that are highly pathogenic. They recognize our basement membrane, for example, in Goodpasture syndrome. They could be very toxic and could be very progressive. And this type of kidney disease patient may have less than half a year remaining. And there are many other autoantibodies that are less toxic. They may be playing a role in disease in a slow process. But I also want to mention that in a situation where these autoantibodies are not directly pathogenic, they could also be an important biomarker to tell us that, in fact, an autoimmune mechanism, a process is going on.
Okay. What did your team of researchers discover about these proteins that are harmful to the body?
Well, autoantibodies are known to be pathogenic in general, but no one has really looked at the population in general, how common these autoantibodies can be detected. And in our study, we use the enhanced samples, which is a very unique collection of individuals supposedly representative of the US population. And usually there are about 5,000 individuals involved in each of these studies. And so by examining this kind of samples, our data represent the frequency of autoantibodies in a representative population. This study established a baseline for autoantibodies in a representative population. So for physicians, they would be able to determine whether other diseases have higher frequency or lower frequency of autoantibodies based on this study. The results were surprising in the sense that autoantibody frequency, although we know is supposed to be increasing with age, but it taper off at about 45 to 50 years old, and especially in women who are more prone to have autoantibodies, they seem to decrease in frequency past 50 years old. The other unexpected result is that normally obesity is expected to be correlated with higher frequency of autoimmune disease. In this case, we found a lower percentage of this individual have autoantibodies. It comes to the important point that autoantibodies can be representative of pathogenic antibodies, but in some cases there have been descriptions that they could be protective as well. So the jury is still out. What are these autoantibodies really representing? I can say for us, the publication is only a fraction of the data analysis of the data that we have collected so far. The enhanced study provides a variety of parameters or variables to examine. For example, how exposure to chemicals and toxins can be correlated with the production of autoantibodies. That's what we will be doing in the future. We'll be also focusing on more specific autoantibodies and examine their frequency in this population. The important point is that autoantibodies are relatively high frequency in the general population, and this is somewhat increased compared to maybe 10-20 years ago. I think this is may correlated with the increased frequency of autoantibodies, increased frequency of autoimmune disease in a population. Whether these are related to the environmental impact that we talk about, environmental impact that has contribution to disease such as autoimmune disease. So we would need to examine autoantibodies in a context of this study. What are the ones that are specific to disease versus those that potentially even be protective to disease?